We will continue with the chapter on infectious diseases. This is AS Biology Syllabus 9700, and this is the second video on this chapter, and in this we're going to cover a little bit of the portion on malaria which was left, and then we're going to do AIDS, and then we're going to do antibiotics. This is the chapter on infectious diseases. Now, if we can recap from the previous video, malaria was caused by Plasmodium, but the insect vector was the female Anopheles mosquito. Now, if we have to control malaria, then we have to control the population of mosquitoes in a certain area if malaria is very common in that part of the world. So we know the life cycle of mosquito. Mostly the eggs are laid on stagnant water, and then after a few days hatch out the larvae. The larvae feeds and the pupa does not feed. So the eggs, larvae and pupa are in the stagnant water. So one way is to drain the stagnant water, which is not possible if there's miles and miles of stagnant water in places where there's a lot of rain. And if there are little puddles of water, yes, we can drain those. So the second method would be that we introduce fish. And this is of course a form of biological pest control because the fish will eat the larvae and the eggs and the pupa. So the number of mosquitoes will decrease. So introducing fish is using one organism against the other, and this is called biological pest control. Then we can spray bacteria which is called Bacillus thuringiensis. This is one word. And this bacteria, when we spray it on the surface of the stagnant water, this kills the larvae. The reason being is the larvae is the only one which feeds. The pupa does not feed. So the pupa will not be killed, but the larvae will be killed. And if there is no larvae, there is no pupa, there is no adults. So Bacillus thuringiensis is a bacterium, and this bacteria is toxic for this larva of the mosquito, but it doesn't harm other aquatic life. So this is one way of controlling the number of mosquitoes. Then we go on to prevention of malaria, and we go on to the three main headings. The first one is reduce the number of mosquitoes. The second, reduce the number of mosquitoes, is the ways that I just told you about the fish and the bacteria story. Then number two is avoid being bitten by mosquitoes. The word bitten is not correct because they don't bite, they just suck the blood. So this is if you sleep under mosquito nets, or if you use some sort of a cream which prevents the mosquitoes coming near you. So those are the factors that we have to remember. Or you wear full sleeves so they don't, the mosquitoes don't come near you and suck your blood. The third point is use drugs to prevent the parasite infecting people. Now this is called preventive medication, in which you take medicines in order to avoid getting the disease. But you don't have the disease, but you're just preventing it in case the Plasmodium enters your body. The next thing is that we talk the topic of why malaria control programs are not successful. Why are they such a sad, tragic story? The reason is that the Plasmodium is the causative organism. That's a protozoa. That's a eukaryote. Why do I keep on reminding you this is because you all forget it. So it's a protozoa, it's a eukaryote. So please remember, it's not a bacteria, it's not a virus. We give certain drugs to control it, and it's becoming resistant to the drugs used to control it. The chloroquine, the different forms of quinine that we use. So it is becoming resistant to that. So new drugs have to be developed, and that is taking its time. The other point which we need to talk about is mosquitoes are becoming resistant to insecticides. You buy a spray, Morton spray, and you say, oh well, this is going to kill all the mosquitoes. And after a month or so, when you use the same spray, you still find mosquitoes around. So they have become resistant. And now some of them have been destroyed by the insecticide, but a few still remain. After another month, those few will now have multiplied and there will be more of the resistant forms. So mosquitoes become resistant. And please remember, mosquito is the vector, is the one which is just going to transfer the plasmodium from one human to the other. Then we talk of the worldwide concern over spread of malaria. The main concern in the worldwide spread of malaria is that number one, there's increased resistant forms of Plasmodium. Number two, there's increase in one form of Plasmodium, which is Plasmodium falciparum. And this causes severe, often fatal malaria. So of course, that means more people will die of malaria because Plasmodium falciparum is more dangerous than the other ones. Then number three, no vaccine has developed against malaria. We have no vaccine which we can give to people and they won't get any malaria. It's a preventive measure. We can't develop a vaccine. Why? Because the Plasmodium is more complex. It's a protozoa and it undergoes many changes in its life cycle. And every time it changes, it has a different antigen on its surface. Then the number of epidemics increased as climate changes favor the spread of mosquitoes. More rain, more breeding grounds for mosquitoes. So this is why we say number of epidemics have increased. And the last point is migration of people due to economic and political reasons from areas where malaria is endemic. Endemic means where people, few people suffer from the disease. Not a large number, endemic. If it's a large number, it's epidemic. If it is in a number of countries, pandemic. We talked about these words earlier in video 1. So when people are migrating due to economic and political reasons from areas where malaria was less, but now they're moving to areas where malaria is more, and so they're adding more people who are suffering from malaria. So it's going to spread more in those countries as well where they're moving to. So these are the preventive and control methods that I wanted to talk about, and that completes the portion which is about malaria. And now we're going to start the new disease, which is AIDS. So we start the new and we'll talk about AIDS now. The letters AIDS stands for Acquired Immunodeficiency Syndrome. The pathogen or the causative organism is HIV, in short, human immunodeficiency virus. Now, before we go on to the method of transmission, I always like to talk of the clinical features. The clinical features in HIV infection is flu-like symptoms, then symptomless, and maybe symptomless for years. They have nothing. They have no fever, nothing. And AIDS results in opportunistic infections because the main reason is where is the site of action of the pathogen. It attacks the lymphocytes, and the lymphocytes are the main cells which make antibodies and destroy the invading pathogen. So if the lymphocytes are destroyed, you don't have an immune system functioning. When your immune system is compromised, then you're going to suffer from opportunistic infections. Opportunistic infection means that your immunity is non-existent. And now pathogens have invaded and have taken over. So opportunistic infections, including pneumonia, TB is caused by bacteria. So people with AIDS will suffer from tuberculosis. And cancers, weight loss, diarrhea, fever, sweating, and in the end is dementia. Method of diagnosis, how do we diagnose somebody who is suffering from AIDS? We'll do the blood test, we can do the saliva, urine for the presence of antibodies against the HIV. So say this is the virus, and the virus has this antigen on its surface, and these antibodies form against it. Now, what we're going to look for is the antibodies. The antibodies which will be present in the blood of a person who is suffering from AIDS. So we're going to look for the antibodies. Now, estimated total 35.5 million, estimated number of new cases 2.3 million, and estimated number of deaths is 1.5 million. It's an estimate, so I just want you to know the figures. You just get to know a rough idea of how many people are suffering and how many people are dying of it. Mostly it is globally, it is found in Africa, Southeast Asia. Now let's look at the method of transmission. The method of transmission is the important, because how does the virus travel from this one person who is the sufferer to some other people? How does it travel from an infected person to a non-infected person? So basically it's in the semen and it is present in the vaginal fluids. So if it's a male, it's in the semen. If it's a female, then it's in the vaginal fluids. And it is usually going to spread by sexual intercourse. And then, of course, we talk of contaminated, it says hypodermic syringes, but I prefer to say contaminated needles, because syringes are not going to really spread it. It's the needles which are going to be coming in contact with the blood. The blood contains the virus, and then we're going to use the same needles on somebody else, and then it's going to be transferred. Then mother to fetus across the placenta. So the virus can cross the placenta, or at birth. When the fetus is being born and passes through the vaginal canal, that is when he or she can get the infection. And the last is mother to infant in breast milk. Now these are nearly the same points that we studied in our O level, so it's nothing new that we are studying in this. So basically I want you to know pathogen, I want you to know the site of action of the pathogen, I want you to know the clinical features, and I want you to know the method of diagnosis. So this is what you need to memorize, and you need to go through it and revise it once again before you go on to the next part of this disease process. Before I continue with the AIDS thing, I think I just missed out this portion on malaria. How are we making improvements in the control of malaria? Number one, we're using modern techniques of gene sequencing and drug design so that we can target specific enzymes and kill the causative organism, that is Plasmodium. The second is the development of vaccines against different stages of the parasite life cycle. So if we can develop a vaccine and we can then give it to people who will then not suffer from malaria. Now, the third most important point is the removal. We find that the poorest part of the world has suffered from these diseases. So in order to remove the burden of disease from the poorest part of the world by donations from wealthy individuals and foundations. So maybe we can help the poorer countries to recover. Number one, because they suffer of the burden of the disease, plus they suffer economically. Because when people don't come to work, then the economy of the country is also affected. So these were the points which I had missed out for malaria. Now we continue with the topic of AIDS. If you look at the AIDS virus, it's very, very interesting to see its shape. It has an outer lipid membrane. Then inner it has the protein capsomeres. And then it has a protein core. And in that we have an enzyme protease. And then this has an RNA strand. Remember, virus has an outer protein coat, and sometimes it has an outer fat or a lipid layer as well. And inside it there is no cytoplasm, and there could be a DNA or an RNA strand. Now, in the case of the AIDS, it's an RNA strand. So we say this is a retrovirus. The other one is called an enterovirus. So it has an RNA plus it has an enzyme called reverse transcriptase. So an enzyme called reverse transcriptase is also present. Now this is very interesting because when it is going to invade a human cell, then this RNA is going to be converted into viral DNA. And the viral DNA will then become part of the host cell. Now, what we need to understand is that this virus, when it attacks a human cell, first of all, what it does is it invades this human cell, and this viral RNA, in the presence of the enzyme, reverse transcriptase, this is reverse transcriptase. Reverse transcriptase. Please remember what was transcription. Transcription was DNA to mRNA. Now here is going to happen the other way around. From this viral, this is the viral RNA. Now the viral RNA, in the presence of the enzyme reverse transcriptase, is going to result in the formation of viral DNA. So this is the viral DNA. And then it's going to form a double-stranded DNA. And then this DNA is going to become attached to the human DNA. Now what is going to happen? So viral RNA becomes viral DNA because of the enzyme reverse transcriptase, and this viral DNA becomes part of the host DNA, and then it takes over, and then it asks the host cell to make more of the protein coat, more of the RNA, more of the protein coat, more of the RNA, and we have lots and lots of virus particles being made in this human cell. And finally, this cell is going to rupture, and these virus particles are going to be released. So this is how a virus reproduces inside a human host cell. Now, if we know this, we can understand how we're going to now treat it. So the different treatments which are possible now are going to be discussed. Now, treating AIDS, we've of course resulted in increased life expectancy. Why? Because we are creating drugs, or we are discovering drugs which are going to just stop the virus from replicating. But we've not been able to kill the virus, or we've not been able to remove it completely. We haven't cured anybody from AIDS. So we are just increasing human being sufferers of AIDS, the dependency on drugs. Second point, the drugs are very expensive. Now the drugs are similar to a DNA nucleotide. Now the drug zidovudine is very similar. It's a nucleotide with the base thymine. Now, if you remember your DNA structure, deoxyribose has an organic nitrogen base, and this nitrogen base is going to be thymine, and it has a phosphate. This is what a nucleotide looks like. So we give them drugs which are nucleotides, but this is a faulty nucleotide. So what happens is, when it binds to reverse transcriptase, it blocks its action, and the viral DNA is not going to be made. So as we just discussed it, viral RNA then becomes viral DNA. So if no viral DNA is formed, the virus particles are not going to be replicated. They're not going to be reproduced. The next point is that this, of course, as I said, zidovudine stops the replication of viral genetic material. So no more lymphocytes are destroyed. And so the number of lymphocytes which are decreasing, that is a benchmark that we use to look at the prognosis of this disease when we get a person who's suffering from AIDS. So we do a blood test and we find out the number of lymphocytes. But when we start giving the drugs, the number of lymphocytes increase, because now the virus particle is not entering other lymphocytes and destroying them. So the fact that the number of lymphocytes increase helps in the immune system, or the immunity of that person who's suffering from AIDS. Then preventing AIDS, can't do much. People educated about the spread of the disease, use of condoms, contact tracing. That is, when we have a person who is suffering from AIDS, we will ask him who are his contacts, has he donated blood. So all these factors will help us in tracing those people who might have also developed AIDS. And then, of course, we do a blood test and find out if they have got it or not. Then injecting drug users are asked to stop sharing needles. Then whenever people are given blood transfusions, we ask them to be screened for HIV so that we do not give infected blood to other people. And that brings us now to the last topic, which is antibiotics. Why do antibiotics kill bacteria but they do not kill viruses? The basic fact is that bacteria is made up of a cell wall. Bacteria has a cell wall, bacteria has a cell membrane, bacteria has cytoplasm, bacteria has circular DNA, bacteria has 70S ribosomes. So what we're going to do is either we're going to target the cell wall or the cell membrane or the 70S ribosome, or we're going to target the circular DNA. But antibiotics will kill bacteria, but they will not kill viruses. The reason why is virus does not have any of these structures. The bacteria has a cell wall, but the virus does not have a cell wall. Virus does not have a cell membrane. Virus does not have 70S ribosomes. Virus only and only has a protein coat and a DNA or an RNA strand. It has no cytoplasm. I keep on saying this again and again because I feel students are not very clear on this. So antibiotics will only kill bacteria. They will not kill viruses. Now let's look at the different ways by which antibiotics kill bacteria. So antibiotics, like we've all had antibiotics at some point, Augmentin, Claricid, Ampiclox, Amoxil. So they are cell wall synthesis inhibitors. They stop transcription. Transcription is DNA to RNA, DNA to mRNA. So we say transcription inhibitors. Then they are protein synthesis inhibitors. So no proteins will be made on the 70S ribosomes. Then they interfere with metabolic reactions. They are membrane inhibitors. The cell membrane, it's partially permeable. If it is made fully permeable, the bacteria will die. So antibiotics work in different ways in destroying bacteria. Now, a person suffering from a bacterial infection is asked to take an antibiotic. And he's asked to take this antibiotics for 7 days. But he or she feels better and after 3 days and stops the antibiotics. You see, when you stop the antibiotics, the bacteria mutates. When the bacteria mutates, there are changes in the DNA and resistance develops. Like for instance, there's an antibiotic called penicillin. Now the bacteria starts to produce an enzyme called penicillinase. And what is going to happen is penicillin is not going to kill the bacteria anymore. Now that bacteria which contains the enzyme penicillinase now is going to flourish and is going to survive. So all the bacteria which arise from this one bacteria now are going to be resistant to penicillin. So bacteria mutates, resistance develops, and all the next generation of bacteria are going to now be resistant to this antibiotic. Now, how can we control this antibiotic resistance? There are a few points which have been taken from your book. We should be using antibiotics only when appropriate and necessary, not prescribing them for viral infections. Reducing the number of countries in which antibiotics are sold without a doctor's prescription. Sadly, Pakistan, you can go and get an antibiotic without a prescription. Avoiding the use of so-called wide-spectrum antibiotics and using instead an antibiotic specific for the infection. Then making sure that the patients complete their course of the medication. making sure that the patients do not keep unused antibiotics for self-medication in the future. You were given a course of antibiotics, you didn't complete it, you had it for 4 days, the rest of it is lying around, and then after a few days, you again take the same medicine because you're not feeling well. Changing the type of antibiotics prescribed for certain diseases so that the same antibiotic is not always prescribed for the same disease. Like, for instance, we give a certain antibiotic for the disease called cholera. So we should keep on changing it so that the resistant strains do not develop. Then farmers use antibiotics to prevent diseases. Like, for instance, they have a poultry farm and they have lots and lots of these little chickens growing, and they give antibiotics in their food so they do not develop diseases. Now, that's not a very good idea. So, for prevention, giving it during farming is not a very good idea. It should be only given when we want to cure an infection which the animals are suffering from. So, these are some of the points that we need to consider so as to help in not developing antibiotic resistant strains. Now, there are two other terms that we use about transfer of plasmids containing the resistant gene: vertical transmission and horizontal transmission. Normally, a bacteria divides by binary fission. So, the circular DNA forms a copy, so goes into both cells, and then the plasmids, which are present, also form copies and they go into each of the daughter cells. So this is called binary fission. Please remember, this is not mitosis. This is called binary fission. So vertical transmission is that when we have a bacteria, which is the resistant parent cell, now this bacteria is going to divide by binary fission, and both the daughter cells are now going to receive the plasmid, which has the resistant gene on it. Sometimes what happens is that this bacteria comes in contact with another type of bacteria, could be the yogurt bacteria. Now this one is the non-resistant one. And what it happens is it forms a conjugation tube, and this plasmid is transferred here. So the plasmid will now enter this other bacteria, which is not related to it, just another non-resistant bacteria. And then, of course, this bacteria will also become resistant. So please understand vertical transmission and horizontal transmission. Vertical transmission is binary fission. One bacteria has it, the offsprings have it. So this bacteria has it, then the offspring, the two that are formed, this one of course becomes these two, so they'll develop it. This is called vertical transmission. Horizontal is when another bacteria, the plasmid is transferred to any other bacteria which it comes in contact with, and the plasmid is transferred to that bacteria. That is called horizontal transmission. The normal procedure for diagnosis of a disease is that you need to grow those bacteria. So if a person is suffering from a throat infection, we grow those bacteria. Now you can see these bacteria growing on it. These are these dots, the green ones. These are the bacteria growing on it. Now what we've done is we placed these discs of antibiotics on it. These discs contain antibiotics. And then after 24 hours, we see this clear area around it. Now, wherever there is the larger clear area, now this antibiotics is the one which is effective. This one was totally ineffective. This one was ineffective. This had smaller clear areas. So wherever the clear areas, that means that when you place the disc, the antibiotic diffuses out and kills the bacteria. But in some cases, it doesn't kill the bacteria. So what we need to do is whenever we prescribe an antibiotic, we need to do this test to find out which antibiotic is going to kill this strain of bacteria. So this is all that I want to tell you about this disease, and please go through this video twice so that you can understand all the different points of this chapter. Thank you.